Hello, everyone. We'll give like 5 more seconds for people to come into the room. So my name is Michelle Gray. I am a assistant professor at the University of Alabama at Birmingham. I am a Huntington's disease researcher. Our laboratory actually generates and utilizes mouse models that express the mutant Huntington protein to try to understand the pathogenesis of this disease. Presently, I'm the chair of the Scientific Advisory Board for HDSA and on the National Board for the Huntington's Disease Society of America. So we're very happy that you guys decided to attend our genome editing CRISPR session today. And so just want to give a little overview, like a quick intro. But not really an intro because Ricardo's going to give us a great intro to CRISPR today. But a little intro, if you can see in your in the guidebook app, but it talks about that over the last few years we've been hearing a lot about the promise of CRISPR technology and how this genomic editing approach could be used to possibly cure Huntington's disease. But as the overview in the guidebook tells you, we're actually a little ways off from that. But we do recognize that this particular technology is very promising and has the opportunity to clearly impact the way we approach Huntington's disease treatment. So we have as our speaker today, Ricardo Pinto. So Ricardo is an instructor of neurology at Harvard Medical School and an assistant in neuroscience in the Massachusetts General Hospital. He is actually a recipient of the 2016 HDSA Berman Topper HD Career Development Fellowship. So Dr. Pinto has a great interest in human genetics and neuroscience with a particular focus on unstable microsatellite associated neurodegenerative disorders, including Huntington's disease and Friedreich's ataxia. Currently, his primary research is focused on identification of genetic modifiers of somatic CAG repeat instability in HD, using HD patient postmortem brain samples, CRISPR-Cas9 in vivo genome editing in mice, and development of novel CRISPR-Cas9 based therapeutics for Friedreich's ataxia. So Ricardo's going to give you a great intro to CRISPR, and he's going to talk about some of the work he's been doing in his laboratory. Thank you.
Thank you, Michelle, for the introduction. So first of all, I want to thank everyone at the HDSA for the invitation, and particularly Michael Berman and Jocelyn Topper for the... Sorry, I think the mic is off. Hello? I just need to get really close. So I want to thank everyone at the HDSA for the invitation and for the opportunity to present here, and in particular Michael Berman and Jocelyn Topper for allowing me to do this research and providing me with the inaugural Berman Topper Fellowship. So I would like this talk to be interactive, even though it's going to be difficult if I have to be this close to the microphone. But anyway, so at least throughout the earlier part of the talk, where I'm going to try and introduce CRISPR, please feel free to interrupt with questions. I think it might be easier so that make sure that everyone's on board and following us. So for that first part, feel free to interrupt. Can I get just a count of hands of how many have heard of CRISPR? Okay, so that's why you came. Okay, so I'll give a little, still, if you haven't heard of it, I've got you covered. I'm gonna try and present that as well as I can. So I'll go over the origin of it, what its normal mechanism and its common use, and why we researchers think it's so powerful and so revolutionary in the field. And lastly, I'll give you an example of a project that I'm working on in terms of how we can apply this tool experimentally. So the key principle behind genome editing is the same thing you do if you have a desk job, is you go in Word and you want to find the word in a document. So that's the key principle, is how to go in book or in a document, find a page, not only find a page, find a word. As you can see, the problem we have in genetics is that the cell, the genome, is a huge book. It's really large. It's got 6.5 billion letters. So it's really difficult to find a single word, and especially when the message is like that, with just 4 characters. So you really need sophisticated tools to go in a book like that, find a page like that, and still find the CAG mutation. So this is what CRISPR has allowed us to do, not only just for HD, but for the majority of genetics, to go into the genome of a cell and target a particular mutation. And this is how it does. So here I'm highlighting Cas9. Cas9 is one of the enzymes under the CRISPR family. So there are multiple enzymes that can have this function. Cas9 is the mostly commonly used. If you imagine a target sequence of about 20 base pairs, there's some constraint in terms of a pan motif, which is next to your target site, and this is required sequence for the Cas9 to recognize and introduce a cleavage. So you need to provide a guide that is complementary to those 20 base pairs, and this guide needs to be adjacent to the PAM motif. The enzyme Cas9 binds the guide, goes and scans the genome until it finds a match. As soon as it finds a match, it introduces a double cut. And interestingly, I've only started hearing about CRISPR in the last 3-4 years, but this is an ancient mechanism that has been used by bacteria to fight infection. So this is bacteria's immune system in a way. So it's an adaptive immune system. Whenever a bacteria, so in this diagram, which I've taken from the Duden lab, you have a bacteria represented and being infected by a virus. Viruses normally take advantage of the DNA replication machinery of a cell to produce more viral copies. What bacteria have learned to do is to take a little fragment of the viral genome and incorporate it into their own genome. And so they keep a library of every bit of virus that they've seen in the past, so that whenever they see it again, they can use this machinery to destroy it, to fight it. So here you see that this bacteria has seen multiple viruses, and so it's kept little fragments of its genome. It has coding region for the Cas protein, so it can make protein, it can make the tracer RNA, combine the two, and then fight the virus whenever it sees it again. So I'll just highlight one thing. What's novel about this is that, you have a question? No. What's novel about this is that scientists have engineered this system so that it's very easy to make this in lab. It's very easy to express this protein. You just need the Cas9 protein. You need to express the guide against a particular target site, introduce it into a cell, and it should lead to mutation. However, there are many things that need to be ironed out here. We still really need to optimize this technology, and there's a lot of effort doing that. Quickly try to go over how this can be applied in the future as a potential therapeutic for HD. And you've been hearing about the various ways of targeting HD and by reducing the mutant huntingtin. So if you have normally every HD at risk or patient will have a normal length allele and an expanded allele. Both make RNA, But this is the driving, there's some suggestion that RNA might be playing a role as well, but particularly the mutant Huntington is what's causing HD. So the strategy is to get rid of this. Some approaches try to handle target the protein directly. Can we prevent it from aggregating? Can we prevent it from having a toxic effect? Other strategies, like you've heard the last couple of days, involve targeting the RNA, and this can be done with shRNAs or with ASOs that either target just the mutant allele or that target both and reduce both the normal protein and the mutant protein. Another strategy is to target directly the DNA. So as you can see, we've been going from downstream effect to all the way upstream to the cause of the disease. And you can do this with zinc fingers, which are another type of nucleases. Here, they've been engineered so that they're not a nuclease, so they do not cut the genome, but they've been coupled with a transcription repressor. So they recognize the CAG repeat, and they silence the expression. So you don't produce RNA, or as much RNA. Ultimately, you don't produce the mutant protein. With CRISPR, all of a sudden, in theory, and this is still very early days, you can target the mutant allele directly. Design guides, so design the system in a way that cuts nearby the repeat in a very specific manner, and either corrects the CAG, and you get 2 normal copies of the Huntington gene, or simply deletes it, and you retain only the normal copy, 1 normal copy of the allele. This is all still theoretical, and it's being done in vitro in cell models. It's being worked out. So here I'm summarizing the 2 basic strategies of how CRISPR could be applied to the clinic, and I'll start with this strategy here, which is very similar to the zinc finger strategy. You can couple the Cas9 with a repressor and use Cas9 to target the promoter of the Huntington gene and silence the expression of the mutant Huntington. Advantage of this, and I apologize, the symbols didn't come out right. This should be good and this should be bad. The advantage is that you do not cause permanent changes, so you're not editing the genome, but it would probably be a transient effect, so you'd need repeated doses. The other strategy, the more permanent strategy, and that would address the underlying cause at the genetic level, is by using the Cas9 cleavage potential, where you can cut around the repeat and excise completely the CAG, or you can cut, again, flanking the mutant allele, provide it with a template, so you need to provide it with a good copy of that, what it should go in there, a normal CAG length, and correct the repeat. The first strategy has been recently tried and actually published by a colleague, John Min Lee at Mass General Hospital, where they've taken advantage of the haplotypes. So different individuals have different polymorphisms. In addition to the CAG repeat, they have different polymorphisms in the CAG gene that would allow you to distinguish the wild type allele from the mutant allele. So what is noticed was that a particular haplotype that is common with the normal alleles has this particular PAM sequence in these sites, which is not recognized by the Cas9. The mutant allele, on the other hand, has a single nucleotide change. So as you can see, this is GT and here is GG. There is CG and here is GG as well. So these would both cut, while none of these would cut. And he has shown this very elegantly in cell lines. When you introduce these double cuts, frequently you introduce errors here, but on occasions you still at a lower frequency, but you get this entire region excised. So you remove the expanded CAG completely. And at least from these experiments, it did not see a single event where the wild type allele was targeted, which is very encouraging. That's the key thing that is important when you're targeting a gene that you're not introducing cuts elsewhere. So at least at these two particular sites, and this hasn't screened the rest of the genome, he's only looked at these particular sites, there was never a single event, where mutations where cleavage was occurring here. So this is still very early days in terms of developing CRISPR tools for the clinic. There are 5 key points that need to be completely improved, and there's a lot of effort from companies that have licensed and developed this technology to make it good enough for the clinic. And we really need to ensure that these 5 points are good. So the first one is activity. You don't want to be giving a drug that is not really cutting the gene. So you need to make sure that Cas9, when matched with a sequence specific to your target gene, has the potential to cut, can cut efficiently. And there have been a lot of engineering around the protein. So even in just the last 2 years, there's been tremendous effort in terms of re-engineering the protein, changing its binding site, not its binding site, its structure, so that it binds more efficiently to the DNA and introduce cleavages more efficiently. The second issue is targeting. So let's say your target is there, but that's got a different PAM sequence. So if you remember, we're restricted by the sequence of the PAM. Say you want to cut there, but that PAM sequence, you don't have tools to target that PAM sequence. Initially, the only Cas9 we had was targeting GG. So we could only cut there if we wanted. There's been a lot of development, and now we've got a selection of different Cas proteins, Cas9 from different bacteria, engineered Cas9s, CPF1, which is a different variant of Cas9, and they all recognize different PAM sequences. So all of a sudden you can target a bigger range of the genome with different enzymes. The third point is specificity. That's your target, but that target is very similar to other targets in the genome. You want to make sure you only cut there. You don't want to cut there or there, because those could be critical genes in your body. So again, this has been re-engineered in a systematic way, and now there are enzymes that have a much higher fidelity. By that, they can detect those small differences, and they will not cut there. They will only cut at your intended place. Again, a lot of this work is still very preliminary, and it's being done in cellular models. So it's impossible to know how any of these re-engineered enzymes will work in an organism. So that work is still being done. The other question is outcome. Cutting is one thing. How the DNA repair machinery handles that edit is a different thing. So there are two main pathways, okay, alluded to very briefly before, to how the cell can repair these cuts. The most common repair mechanism is non-homologous end joining. So you introduce a cut in your target site, and what the repair system does is try to stick it back, and normally it will either delete a few bases or introduce a couple new ones. So what you have here is normally a message lost. And interesting example. So if you imagine these words, Huntington's Disease Society of America, if all of a sudden you introduce a couple of letters there, it doesn't make sense. So you're losing the message here. This is, as George Church calls it, who is a famous geneticist and one of the pioneers of CRISPR technology, this isn't really gene editing, this is gene terrorism. Because we're not really cutting it precisely. We do not control the message that gets integrated. Could be a message lost, it could be a message gain as well, a message that is toxic. So this has that caveat. The other mechanism is homology-directed repair. So you introduce the cut and you provide it with a template, something that you want to go and replace that information that was there, and you get the message corrected. The problem right now is that the mechanism on the left is still, the cell prefers that mechanism than this one. So there's a lot of effort in the field to try to make this more efficient, as opposed to just joining it back up. The other important issue is how do you get this to the right tissue? One thing is us working with this in cells in the dish. How do you get to the brain in a mouse? How do you get to the brain in a patient, or the liver, or any other tissue that you want to have a therapeutic effect on? And here I'm showing GFP expression in, I don't think you can see much, that's because there isn't much signal. This is using the best AAV9 delivery vector to the brain of mice, and normally you need to inject it into the brain directly. This is actually an image of an injection systemically, and this is a new vector that Ed alluded to yesterday. So this is work done by Ben Deverman, and they've performed a screen on modifications of that vector, and they've developed a new one, which they've named PHPB, which, as you can see, even injection systemically, so just into the vein in the tail of a mouse, you get an infection of most neuronal cells in that mouse. And this is an adult mouse. Even afterwards, the blood-brain barrier has been formed. So this is there's been a lot of effort, and I think we're starting to get some of the tools to get these enzymes, these tools directly into the tissue that you want to treat. Questions so far about the technique, the mechanism? Because I'm going to transition to applications, how I'm using it in the lab myself. And so if you have any questions specifically for the mechanism, please feel free to ask them now. Shall I go ahead? Okay. So gonna highlight a question that we have that is quite a difficult question to address, that CRISPR technology has really allowed us to start thinking about ways of understanding and getting a better message. And all starts from the strong dependency that you get between the length of the repeat and the age of onset. So with larger repeats, patients tend to develop symptoms earlier, but also the fact that there is still a tremendous variability. For a particular CAG size, the onset of symptoms can be separated by as much as 20-30 years. So only 70% of this variation is explained by the CAG. There's evidence that of that unexplained variation, so of the remaining 30%, 40 seems to be heritable. So these suggest that there are genetic components that contribute to how early or how late you end up developing HD. And a couple of years ago now, a study led by Jim Gazelle and Marcy McDonald at MGH, which took advantage of a large cohort of samples, about 4,000 patient samples of European descent, and this is important because we're doing a genetic study. You need to make sure you normalize, you minimize the variation between the individuals, and also with a narrower CAG range. And what you do in a study like this is you genotype these individuals at various points in the genome. And then you ask, does the presence of this particular variant, is that associated with an earlier or with a later onset of CAG after correction for the CAG length? So you take into account the CAG length and see how much earlier or later than expected did you develop the THD? And this study identified a few significant trails which are now being followed up on. But a particular trail that we're very interested was this, which didn't reach genome-wide significance, but this is a gene that we had been working on and that we stumbled upon quite a long time before this study. And that came from some mouse studies that we have done. So we had knock-in mouse models in different genetic backgrounds. So imagine the mouse models in different races. So they had different genetic background. They had a comparable CAG, but they had a different genetic background. And what that did is that it altered the somatic CAG instability. And so somatic CAG instability is, even though you inherit a particular CAG size from your parent, in somatic tissue, so in certain cells of your body, that repeat still tends to expand with time. So there's a strong bias for it to expand, and it's very CAG length dependent. So the longer the CAG, the more prone it is to expand with time and with the life of the patient. So this is looking at mice in the striatum and in the liver, and they're about 3 months old here, fairly young, but there's already a significant degree of instability, as you can see here. So the black 6 mice, which is one of the strains, have more instability than the 129 mice. And just to explain how we measure this, so if you imagine the HTT gene, the exon 1 and the CAG mutation, which has a variable length, we have primers around it with a fluorescent tag. And if we do multiple rounds of amplification of PCRs, so to try to probe all the different HTT alleles in a population of cells, so if we extract the entire striatum, what's the distribution of alleles in the striatum of that mouse? And you will get a variety of different products that then you can measure their frequency. So this is the main size that was inherited, and you can see that Here you've got a population of alleles that has expanded from that length. So knowing that these two strains have different phenotypes, so they have different instability profiles, we designed a study where you cross the two strains and then you further intercross them so that each mouse is going to be unique, is going to have a unique genetic signature. They will all have the same HD mutant gene, but they will all have a different genetic nature. And once you genotype them for variants that distinguish the two strains, and you quantify the instability, you can then do again a linkage study. So here, representing the instability levels of the black 6, the 129 much lower. These would be the F1s, so suggesting a dominant effect. And when you have the F2s, all these mice down here, you have a big spread of CAG instability scores. So different degrees of instability. And with, say again? This? No, this is a striatum, sorry, yes. So this phenotype, this screen was done in the striatum of the mice. So the tail is very stable. It doesn't allow us to, even throughout the life of a mouse, up 1 year, 2 years, the tail changes very little, while the striatum is very significant. Even after 2 months, we start seeing significant changes. So this is a metric that measures the spread of CAG. How much has it changed from the original allele that was inherited? And by a method that I'm not going to try to explain because it involves a lot of statistics, but we basically identified a single region in chromosome 9 of the mice, which was on top of an MLH1 gene, which is a mismatch repair gene. So a gene that goes around the genome and checks for errors and tries to fix them. And as you can see here, if you color code those same animals, so each dot is an individual animal, I just want to clarify that, and these are the same animals as you have here, but here I'm color coding them based on the genotype for that particular gene, for the variant. The blue means they have 2 copies of the black 6 gene, and they're all up here. The green means they have 1 copy of each, and they're also up here. But when they have 2 copies of the 129 line, and if you remember the 129 is the one with a lower instability score, they're all down here. So we're very confident that we were very confident at the time that this was the gene underlying this expansion mechanism in these 2 strains. So next we cross these mice with mice that do not express that gene. So if you have lines that don't express MLH1 gene, what happens to the instability phenotype? And interestingly, so we've got tail samples and striatum samples, mice that have 2 functional copies of MLH1, mice that only have 1 functional copy, and mice that have no MLH1. And what we see is when you remove this protein, you completely suppress instability. And this leads to nuclear Huntington accumulation. So the mutant protein is also accumulated less in the nucleus of these cells, of these neurons. So this is very interesting, and we already had shown that other mismatch repair genes played a role in somatic instability. But these are critical DNA repair genes. For example, MLH1, if you turn it off, these mice will develop cancers early in their life. So mutations in MLH1 have been frequently described in patients with colon cancer, so familial disease, Lynch syndrome. So this is not really good therapeutic target, we believe. But it points to a mechanism that we think has some therapeutic potential. And you might be asking, so the mice have somatic instability. How relevant is this? Do HD patients have somatic instability? Does the repeat that they inherit and that they get measured in their blood, is that the same as what's in a neuron? And the answer is no. So this is some old data from Peggy Shelburn's lab, and this is southern blot. So looking at HD alleles in brain samples from post-mortem brain samples from HD patients. And here you can see this is an individual with a very large repeat, 80s, but this already got a dramatic spread of expansions. So this is normal size, and this is going up. So the larger they migrate up, the larger the repeat it is. And here we see it very significantly in the cortex, but not so much in the striatum. But I'll just remind you that this is an end-stage patient. By this point, most of the striatum has been lost. So we're not really looking at neurons here. There's hardly any medium spiny neurons left in this brain. And interestingly, what they've shown as well was that from a couple of samples of individuals that had died not from the course of HD, but from other reasons and prior to any neuronal loss. So even before they started losing any neurons, they passed away, and that sample was analyzed. And here you can see that in the striatum of those individuals, even though they had only 41 CAGs, there were some very large expansions in those neurons, as high as 1,000. So we do. So it's very difficult to link this, because it's extremely difficult to track the size of the repeat in the brain in a living patient, right? We can do these studies post-mortem, but it's very difficult to track how this is contributing to the disease. But still, this uncanny association of the relationship between the length of the CAG and the age of onset, the fact that you're seeing these really large expansions in the tissue that is the first target and the first place where you get neuronal loss, makes us think that this is contributing to that mechanism. So if we can prevent these alleles from jumping up, we believe that that might be a therapeutic, a viable therapeutic strategy. Question is, we really need to understand how that works. We want to prevent that from happening, but without causing any other types of mutations. An earlier study trying to suggest the relationship between the magnitude of these expansions and the age of onset was done by my postdoc mentor, Vanessa Wheeler, where she quantified instability in the cortex of 24 individuals that had either very early age of onset or very late age of onset. So they were all normalized for the CAG length, but they sit at the extremes under this curve. They were either very late or very early. And what was observed was that the individuals, and for comparable CAGs, the individuals with earlier onset had a higher frequency of these expanded alleles. Again, this is an association. There's no causality here. It's just an observation. But it fits a very sounding hypothesis, at least in my opinion. So we know already from these mouse experiments that if you knock out MLH1 and if you knock out one of its binding partners, MLH3, you completely suppress instability. And we know from previous studies that if you suppress MSH2 and MSH3, you also have the same effect, exactly the same effect. So we believe that what's happening at these CAG repeat loci is that these are not normal repeats. The fact that you're going through a repetitive region, it's very prone for secondary structures to form. So what we believe is that you have these tiny hairpins forming at the CAG repeat that gets recognized by the DNA repair machinery as a mistake. There's something wrong here. So there's recruitment initially of MSH2 and MSH3 that binds to those hairpins. Binds those hairpins, then it further recruits other components of the mismatch repair pathway. We have evidence for MLH and MLH3. There's some suggestion that PMS2 might play a role. The fact that in the mice, the MLH3 has such a drastic effect makes us think this is the main driving force behind the instability. But still, we don't know what the mechanism is. We don't know of any other elements upstream that might accelerate this or other elements here. And so just say from a point of view of this being a therapeutic target, even though MLH1 and MSH2 do cause cancer in these mice, and there's a lot of patients with colon cancers with mutations in these genes, there's no reported mutations or no association with cancer for mutations in MSH3 or in MLH3. So suggesting that this might be possibly explored as therapeutic targets. But in addition, as I was saying, we really need to understand how this plays out. What other genes contribute to this? And are there safer genes that we can target in this pathway and prevent somatic expansions from occurring? So with that in mind, and this was the scope of the proposal that the HDSA has funded, it's taking advantage of mice that already have been engineered in a way that they express Cas9. So we don't need to deliver Cas9 to these animals. They already express it. They are healthy, surprisingly. The fact that that's not necessarily surprising, but it was a good finding to show that these animals, even though they have a nuclease, a foreign nuclease constantly being expressed, they don't have any side effects. So it's suggesting that you only start having cleavage or any non-specific effects once you start providing the guides, the other element. So they have one component of the CRISPR system. They have the enzyme. Now we need to deliver the RNA component, the guide, the sequence that is going to specify the target that we want to turn off. So we want to use this system to deliver into these mice either by AAV8 into the tail to target the liver, or originally with AAV9 to target the brain, but they're now considering PHP, potentially systemically. So we want to deliver guides against all these candidate genes that have been identified from the genome-wide association study in patients. So genes that have been shown to have an association with the age of onset in patients. So we want to study those, but also our understanding of DNA repair machinery. We wanted to test other components and see their contribution to instability. So the idea is that if you inject and then you treat these animals for a month or two, and then you collect, you sacrifice the animals and you collect the liver or the brain, depending on what you're targeting in that experiment. Initially we're targeting the liver because that is just easier. But so let's say this is the profile that an untreated animal would have. If you observe that with the targeted gene, you probably that gene has no effect to that phenotype, so you're not changing that. if you end up having more somatic expansions, that's also interesting. You're also identifying a gene that is normally protective. So this is all knockout strategies. Turning off genes that might play a role. So if we see an increase in expansion, it means that gene is good. It's protecting it from expanding. And also we want to identify things that are bad. So when we turn them off, we stabilize the repeat. So I'll just describe more or less the design of these experiments for a particular guide. These initial steps these days are fairly easy for any molecular biologist. There's been a tremendous effort from the CRISPR pioneers to make these tools available to every scientist in the world. So these days it's fairly easy to anyone with a little bit of molecular biology knowledge can turn off a gene in a dish. So we start off with the sequence of the gene that we want to target, and we design what each dot would be a guide on different elements of that would fall under either different elements of that protein. And this is based on prior experiments and computational models. You can predict the efficiency of that gene. Not only the efficiency, but also minimize its off-target effects. So you'll get here a score of how good that guide is. So we select the top ones, and also we select the ones that fall on the earlier part of the protein, because if you tend to introduce a mutation at the end of a protein, sometimes the initial part of the protein, it still has a function. So we want to make sure that we completely eliminate the full-length MLH1 protein. So we introduce these guides in combination with the Cas9 in cells. So the initial validation is all done in cells because that's much quicker and much cheaper. We introduce this in cells, and then we measure that site. How often are we introducing mutations? And we can see, for example, here I'm comparing two guides, that one, and you can see that with this particular guide, we've got as much as 80% of the alleles are being edited, which, given prior technology, like 2-3 years ago, this is remarkable that we can do this so easily. And in terms of validation, so we're looking here at the Western blot, so measuring the amount of protein that is expressed with and without treatment. So here we show samples from mice that have the gene, that have expressed that protein, and mice that don't. So we know that this is the specific MLH1 protein. And here are our treated cells. So untreated, so you still express large amounts of MLH1. And with both guides, even with the one with lower editing potential, we're significantly reducing the amount of this protein. And this is, as I alluded to first, this is the genetic terrorism part. This is turning it off. We don't care how. And we're just using this as a tool to conduct our experiments. We're not trying to design a therapeutic. This is just an experimental tool. So there would be a variety of different mutations that we're introducing. What we care about here is that we're getting rid of this protein. This is some old school Sanger sequencing to try to capture some of the information that's going on at that target site. And this is after taking those two same guides, A1 and JD4, both targeting MLH1, and this is the region in the genome that they hit. And this is, we've cloned them, packaged them into AAV9, and injected them into the Cas9 expressing mice. And as you can see in these highlighted yellow boxes, so this particular mouse was treated with just the A1 guide, and then we're sequencing the A1 target site and the JD4, and you can see there's no mutations introduced, so the sequence looks clean at the JD4 site, and as you can see there, the sequence in the yellow boxes goes out of sync. That suggests that you're introducing indel mutations. So this is just a visual readout. So we're really confident we don't have any mutations when you untreat, and these are very specific to the guides that you're using. And this is just a different way of quantifying that. This is when we do deep sequencing of those target sites. Again, the same mice when looking at one site or the other, and this is the treatment. So for this particular site, if we treat with A1, we get about 50-60% cuts or mutations. So we're editing about half of the alleles in the liver of this mouse, and we're not touching the other target site, and vice versa. When we treat these animals with the other guide and we measure the respective target site, we're getting as much as 70-80% of alleles being edited. So we do believe that we've got a good system. Even with these sort of efficiencies, we have ways of selecting just the green bars. We can isolate just the edited cells and then measure instability there. So this is just a proof of concept that we're doing the follow-up experiments now, where we're turning off these genes in HD mice that express Cas9. And so going back to the other diagram, when we mutate this gene and other candidate genes, do we affect that instability profile? Do we suppress somatic expansions? Do we aggravate them? We want to understand that mechanism completely. Here is just trying to highlight some of those pathways that we're targeting, or that we want to target. Some of them implicated previously in mouse studies, in patient studies for association with age of onset, and others simply because they are in pathways that we think would have an impact in instability, and we'd like to really tease that out. So I want to thank Tonio, who's a student in the lab and who's been helping me over the last year and a half. Vanessa Wheeler is my postdoctoral mentor, and everyone in her lab. John Min, Marcy McDonald, Jim Gazella at CGM. HDSA for the funding, the Harvard Neurodiscovery Center for the funding, and as well as CHDI. And can take any questions. Any genetic disease, in theory. So there's a lot of effort for using it for cancer, for immunotherapy. The easier diseases that are going for are from easier to access tissues right now. So for blood diseases, liver diseases, macular degeneration, for example, things in the eyes, things that are easier to access. In theory, any genetic mutation can be fixed. And again, this is still very early stage in the development of the disease. The companies that are working on this, they still have 1-2 years of experience in the field, so there's still a lot of work that needs to be done. It's moving extremely fast, the field, so that's encouraging, but there's still a lot of work that needs to be done. Yes. Yes.
Yes,
My understanding is that they're already going on in China. So the question was, how long are trials, inpatient trials, going to start? I think it's very far from. I can't comment on a timeline. That these information is still fairly well guarded behind some of these companies, and I don't think they know yet. My understanding is that in places where it's not so well regulated, some of these tests are already being done with potential unwanted consequences. Yes.
What's the time frame between injection and testing?
So these particular experiment that I was showing the results, that was just 8 days. So the question was, what's the time frame between delivering these construct and measuring the effect, the gene editing? So in cell lines, we normally check that 2-3 days later, and we already see that it's really a quick effect. In these graph I showed from the only in vivo data we have so far in mice, that is only 8 to 10 days after injection, and you already have those high efficiencies. We don't really think they're going to get higher. I think it matters that if the timing is quick, is what proportion of cells do you get infected with your construct. We haven't looked. We haven't looked at that yet.
Specific guide RNAs, and is it a problem that the repeat is unstable? So if we're going to cut the repeat out, so is it a problem, or are those sequences that flank that enough?
I see. So the question is, taking into account the fact that the repeat is expanding throughout the life of the patient, and that the easiest way of fixing that problem is by cleaving with CRISPR, flanking the repeat. Would that be interfere? I don't think so. Potentially. So some of the experiments that were done were deleting very large chunks of the genome, something like 40 kb. So a repeat that is as long as a couple hundred bases won't probably have that much of an impact, or even if it goes into 1,000. Yeah, correct. So there are different strategies, I think, and what is been tested so far is using guides not targeting the repeat, but flanking the genomic region around the repeat. So the strategy used by John Min is taking advantage of the haplotypes. So these polymorphisms that are different in one allele versus the other, and that would affect whether it cuts or not. So that technically, you can be as close or as far as possible from the repeat. But it still needs to be optimized. The rate of non-homologous end joining. The HDR. So not the end joining. You want to minimize that and improve the homology-directed repair.